All right, thank you very much. So yes, I'm going to talk about some quantum biosensing today, work that we've been doing and in collaboration with Pilgyu and some other members of the bioengineering department, as well as physics. So I'm just going to pretty much give an overview of quantum biosensing, and then I'll hand it off to Pilgyu. I'll talk a little bit about my research as well. So to begin with, why do you want to go to quantum sensing in the first place? What are the hurdles that we have these days for biosensing? What I'm interested in is epilepsy and the study of the human brain. So there's some very specific problems with sensing from a brain. But in general, what we want is to have a sensor which is obviously sensitive. So you want to be able to pick up very small signals, which is important, especially in things like the brain. But you also need specificity. You don't want to have, say, glutamate and, say, dopamine and have them both give you the same signal. That way you can't tell which one is which. So the combination of sensitivity and specificity is always important for sensing. Now, for biosensing, another thing that's important is targeting. So you'd like to be able to get your probes to go to the place that you want them to go. And so how do you do that? What are you going to do to get that probe there? Can quantum do something to help us in that regard? Can quantum do something to help us in sensitivities? Can quantum do something to help us with specificity? Then there's attenuation. That is how far through the tissue can you look? So typically you either use light or you use magnetic or electric fields. Is there some way that we could use some kind of quantum interaction, some kind of spooky entanglement to be able to really be able to go deep into the brain? And then clinical, is there something that will enable us to make measurements that we can do in the laboratory, but we can't really do in the human body? Is there something that could be offered by quantum to help us to overcome that hurdle as well? So I'm not going to address all of these things. I just wanted to throw out there kind of a motivation as to why we want to use a new type of probe. So when you think about biology, you're really thinking about cells. You're thinking about animal cells in general, although plant biology is very interesting as well. And if you look at an animal cell, it's very complicated. There's a membrane that really encompasses everything inside, and then inside there's lots of organelles and different structures. Do you want to go measure from them? Do you want to go to the nucleus and see what's going on inside the nucleus? So these are all questions you might want to ask if you're going into biosensing. And so one of the most important structures is the membrane of the cell itself. The membrane is what allows things to come in and out of the cell. And if you're talking about neurons, the membrane is what transmits the signal. So a neuron on the left is a schematic of a neuron. A neuron will produce what's called an action potential, which is a voltage fluctuation which propagates down the axon to the next cell, and then at the end of the cell, it will release some chemical transmitter that will be received by the next cell. So communication occurs in this manner, and it really is driven by the dynamics of the membrane. So you'd want to get some kind of a probe that can go to the membrane, find some aspect of the membrane, and attach to it. Like I said, my personal interest is in epilepsy. It's a chronic disorder. There's actually 40 different disorders that go under the heading of epilepsy. It's generally, the onset is generally either with children or with older age. And that's because there's kind of two big types of epilepsy. There's what's called idiopathic, where we don't really know the actual cause of it, but it's assumed that it's some kind of a genetic mutation, which has led to some kind of change in the protein structure. Or symptomatic, which has to do with something that you can actually see. Is there a lesion? That means, is there a scar in the brain, or is there a tumor or some malformation? And so idiopathic generally shows up early on in life, and symptomatic is more of an acquired type of disease. But whether you look at idiopathic or symptomatic, about a third of people that have epilepsy can't use drugs to control their epilepsy. So we need to have some other way to deal with that. And so sensing, being able to see what's actually going on inside the brain, being able to understand the dynamics of epilepsy is really my main focus in my research. And so I said, what are the dynamics of epilepsy? Well, there really isn't one. There's many. And so you can talk about I wouldn't call them benign. They can be quite dangerous, absence seizures. They're not very dramatic. If someone has an absence seizure, you don't really even know it in general. Or a grand mal seizure, which is the typical kind of seizure that people think about, where someone will lose consciousness and they might have a lot of muscular movement as well. So if we want to study those, we need to try to understand what kind of basic dynamics go on, and then how do those change for these different types of epilepsies. And here's a picture of a hippocampus from a rodent. And so all the different colors here are showing you different cells. And I just wanted to throw this up there to show you, it's not like you've got a bunch of neurons sitting around. It's packed. It's really a tight situation. If you want to get probes in there, you need to make use of the vasculature, of the blood vessels. You need to, which are all throughout here. You need to use those blood vessels to take your probes in and then deposit them where you want to measure. This is what neurons look like. If you can stain just a few of them, this is from an in vitro, meaning it's been removed from the animal. And here are what seizures look like in an in vitro setting. So again, this is a slice from a rat brain, and these are what seizures look like in those slices. So what we really want to do is not just do slices of brains. We want to look at the brain, and we don't want to look at the brain removed from the animal. We want to look at it in the animal as he's moving around, having a seizure, figure out how to measure when he's going to have a seizure, and then do something to stop the seizure. The one thing we found in our research, which is very important, is that, and I won't go into the details of this, but potassium and sodium are the two ions that are responsible for neuronal signaling. And in seizures, what happens is that you get a very large buildup of potassium outside of the cells. And so I only bring this up because what would you want to measure? What do you want to look at if you want to see what's going on for seizures? Of course, electrical activity is important. You want to measure those electrical signals that go down the neuron to the next neuron. But you also want to measure things like potassium. They tell you when things start to get a little bit twitchy, when things start to get a little bit too excitable. All right, so just in general, if you're studying biology, again, I'm kind of influenced by my study of the brain, so a lot of this has more to do with specifically the brain. But in general, there are three big categories of things: ions and simple molecules, and then signaling molecules, which are kind of intermediate in terms of their complexity. Those are like dopamine and say serotonin, and then big molecules like proteins. And so if you think about trying to study things like viruses, you'd be wanting to look at proteins. So these are kind of the big groups of different types of molecules that you would want to be able to target. So how are you going to target those? Well, biology can help to some extent. We can manipulate biology. We can manipulate the code of an animal and get it to actually fluoresce. It can produce proteins that you can use to look at different cells. This is what's called the rainbow mouse. And so all these neurons, all these little colored things are different colors due to their genetic code. And so you can do this with just staining so that you can see which neuron's connected to which other neuron, or you can have them produce other more interesting indicators that can tell you how much calcium there is inside the cell. And calcium is another indicator of activity. All right, now typically what we do when we want to study the brain is we use chemistry. If we want to use a sensor, most sensors these days are based on chemistry. And the chemistry obviously is quantum too. So this is some pretty interesting quantum behaviors here if you talk about fluorescent molecules. So in the middle here is a commercial molecule that I use for studying oxygen. It's called platinum octaethyl porphine ketone. And if you look at it, it's got a nice kind of structure. It's beautiful. All these are really cool to look at. If you look to the left, you'll see that there's a very similar structure. And it really is based on the same porphyrin ring. So that big structure in the center is really similar between the two. And the molecule on the left is heme B. So that's in your blood. So that's what's responsible for carrying oxygen around through your body. And so it also fluoresces. So blood fluoresces. You can use it as an indicator. And on the right-hand side is another molecule made by a bacteria that you can use for measuring potassium. And here's some results that you can see from studies that we did where we were looking at a brain slice, and we were using probes with that center molecule from the previous slide to look at oxygen concentration. And we were able to reveal using this probe that there's a threshold at which if the oxygen goes lower than that, seizures will be initiated in these slices, which indicates that there's some metabolic constraint, that if you go below some ability to produce metabolism, then you might have seizures. like I said, electric fields are the other thing that you'd really want to measure. And so there's ways to do that again with dyes. And so there's a large category of molecular dyes that will go into that membrane, and then they'll change their fluorescent properties. Either it can be by having the excitation shift, so the light that it absorbs, the quality, the color will shift either to longer or shorter wavelengths, or the fluorescence will shift to longer or shorter wavelengths. And you can use those to measure the voltage. On the right-hand side is a commercial dye called Fluvolt, and I've changed the voltage with a patch clamp, and you can see that it goes up and down in response to me changing the voltage. And so these are nice dyes, but again, these are molecular dyes, and they're pretty standard in biology these days. So quantum, what can we do with quantum? What has been done with quantum? A bit, not a whole lot. One of the first things that's been done is using carbon nanotubes coated with a polymer, and the polymer makes the carbon nanotube specific to different molecules. So you can put things like these fullerenes, these C60s, into the carbon nanotube, or you can coat them with some kind of a surfactant or some kind of a polymer, like I said, to make them functionalized. Carbon itself is just going to absorb and not really re-radiate it, except for in the black body. It's going to get hot. And that's great if you're going to do some techniques, but for fluorescence, it doesn't do anything. You see nothing. But if you functionalize it, you can change the states of the carbon nanotube so that it does fluoresce. So if you look here on the right, here's a nice result where when you add dopamine, it's actually a turn on sensor, so that it's dark, and then when dopamine comes and binds, it turns that carbon nanotube from just an absorber to an absorber and a fluorescer. Here's some work that Pilgyu did, looking at producing graphene from other materials using heat. He might talk about this, I'm not sure. I just wanted to throw it out here because this is another way that you could functionalize a quantum-based material to potentially monitor either nitrogen or temperature, glucose, or different ions. Or we can talk about even more exotic materials. And I just threw this up here because it's cool. This is material that will maintain a spin texturing. I'm not saying that this would work for biology. I'm just saying that if you can produce some kind of a structure in your quantum material, maybe that structure then could be used to sense some type of molecule. So I'm just throwing it out there. I'm really on the biology side. I'm trying to bridge the gap between the quantum materials and being able to use those, but I'm really relying on the quantum folks to tell me exactly what their materials might be able to do. So again, what can quantum do? Perhaps it can increase the sensitivity, the specificity. Can we use it for entanglement? Probably not, because the temperatures are too high, but it's always something interesting to think about. Another thing that might be interesting to try to utilize is energy harvesting. So you have a lot of energy in biological systems. And so could you make something is something like a photodiode or an LED that can use the energy around it to produce its own signal? So you don't have to shine light on it. It just shines light out just by scavenging energy from the surroundings. All right, so I better move pretty quick here. So how do we make a probe? So we can make some materials, some quantum materials. How could we get those materials into the brain? So I'm collaborating with a bioengineer, Remy Veneziano, who does a really awesome technique called DNA origami. And so what you can do with DNA is you can come up with a sequence of DNA, and the DNA then will just fold up into a structure. You have to do nothing except for come up with the right sequence. It's easy to make these sequences, and you can make all sorts of cool little structures. So you can make rods, you can make pyramidal structures, you can make little balls and different, whoops. All right. And so you can make all different types of little structures with those. So what's the point? What the point is, you can take those structures and you can have them bind to things. You can have them bind to dyes. So we've done this where we've taken a dye called ICG, which is a voltage sensing dye, and we've combined it with these DNA scaffolds. And Remy's also been able to put on these DNA scaffolds other molecules that target. So they'll go, and not only will they find a neuron, they'll find a specific part of the neuron. So maybe they'll go to the soma, or maybe they'll go to the axon, or maybe they'll go to the dendrite, so that we can get very good information about what's happening in different parts of the cells. So we've done this work. Here's some results. I showed you the flu volt earlier. That was a nicer result in terms of its reliability for reproducing the voltage signal, but we're still getting very good responses here using this construct that Remy made. So just the next step is to try to use that same DNA structure, not use dyes, but use some quantum materials, use some kind of fancy new quantum type of material, and put that, conjugate it with our DNA scaffold, deliver that to the brain, and make measurements. And so this can be done. You can take all sorts of different types of 2D materials, grind them up into small pieces, and then potentially put those into the brain. On the right is some work that Pilgyu and I have worked on recently, where I've plated some HeLa cells onto a little pad of graphene. So you can see on the right side, it's called a bright field image, where you can see in black the little pad, and then the green are fluorescence of the cells that are there. So that's just to show biocompatibility. This is a big macroscopic pad. The idea would be to go the other direction and break it up into small pieces so that you can deliver those tiny little pieces through the vasculature to neurons in the brain. And this is this requires a lot of collaboration. And so this work is done with bioengineers, with physicists, and we also work with mathematicians and psychologists who study the brain to get this work done. So I'll hand off to Pilgyu now, and I'll allow, if you want to ask any questions, I'll stick around until he's done.
Thank you so much, Rob. And just to moving on, just I hope I can share what we do in our lab, in our sensing groups, and then how we do with nanomaterials we deal with. So, Dr. Cao, how much time do I have?
15 minutes.
15 minutes? Okay, all right. So let me share my screen. I'm not going to just make all the details, but I'm just giving you a sense that what we do and how we do. So in our lab first, our lab is located in IUVR, Institute for Biomedical Research Centers in Manassas campus. And what we do is we synthesize atomically thin materials. So you may have heard the 2D materials of the graphene. Graphene is one of the representative materials, atomically thin materials, just one thin sheet of the hexagonally bonded carbon atoms. And then we synthesize those materials and we use that for biomedical sensors. And we work with the people in different groups and different institutions, Cornell's and NIST, and also I'm affiliated in the quantum science and engineering centers, and then we collaborated on many projects and interesting projects that I'm gonna introduce today, but I'm gonna give you a sense of what we do. So first, I'd like to just introduce what we do. In our lab, we synthesize materials using different techniques, the chemical vapor depositions and then laser manufacturing techniques to make these atomically thin materials. So using these materials, what we do, we use these materials by, making nanostructures, very small scale structures. Even with these atomically thin materials, we create small structures and then modulate the material's property. I mean, one of the key characteristics of these atomically thin materials, they have a very exceptional electrical and mechanical properties. For example, like a graphene, people consider it it has very high mechanical strength, because metal, for example, they get fractures, and even with just a few less than 1% of the strain, like if you stretch it and it gets fractured. But the graphene, is considered to have a very high, mechanical strength. Even at like 2 to 3% is exceptionally high mechanical strength. And but as we make nanostructures, even we can enhance those mechanical strength, and at the same time we can also enhance the electronic property too. The graphene, for example, it has high electrical mobility, so it has high conductivity, even though it has just it's just one inch of just hexagonally bonded carbon atoms. But as we make structures, it shows interesting properties. So we use those structured atomic lithium materials to make nanobiosensors. So just to give you a sense of what the atomic lithium materials, it's also called the 2D materials. It's just one or few atom-thick materials of the thin sheets of the atoms bonded by those, covalent bonds. And the graphene and MOS2 those are the representative materials that people use widely for broad applications. But as we create those, small structures, even with these very thin atomic lithium materials, we enhance the material's properties and also create some interesting functionality. So that we're going to talk about. So first, let's talk about how we synthesize those atomic lithium materials. So in our lab, we use a chemical vapor deposition tool. So this is a tool we currently have in our lab to synthesize large scale. You can think of a 5 by 5 centimeter scale. Using the catalytic film, we can synthesize those materials with the high temperature, like 1,000. Using methanes and hydrogens and argon. Those gases can be a precursor. So in a very high temperature, up to thousands, those methane gas decompose into carbon and hydrogen atoms, and with the copper atoms, copper film, the copper absorb only the one carbon atoms on the surface of those copper films. So using the absorptions of, just one atom of the carbons, and we can, make the large scale 5 by 5 centimeter size, the carbon, hexagonally bonded carbon atoms, and they are the graphene. So using high temperatures and diffusion of carbon atoms to form these hexagonally bonded structures, we can synthesize large-scale graphene films. And using those, first we characterize how much quality we get. So in terms of the crystallinity, how high quality of the hexagonally bonded carbon atoms we can create. We characterize the quality of the materials using these proof stations, and then we measure the conductivities and electrical property of the material using the probe stations. And we characterize also the optically, so by looking at the material's properties using the scanning electron microscope or optical microscope. We first inspect the material's quality and structures in a micro or nanoscale. Here, using these optical microscope, and we can see even the micro scale structures. So, and also we fabricate the device using these nanowire bonders that we have in our lab. We can make devices and to use for the nanobiosensors in a bio application, for example. So after we synthesize materials, and I mentioned that we created nanostructures. So using simple techniques here, it's called the Shrinkamen of Manufacturing. So we can create a nano or micro structures using this technique, shrinking. So here, first, you can imagine we transfer these graphene on a flexible substrate, and then first we stretch the substrate and then transfer. As we release these stretching, then what happens is we get these wrinkles with these graphene, atomically thin sheet of the graphene materials, and then that way we can create these small nano or micro scale wrinkles, and this is how it looks like. So as we use these shrinking nanomanufacturing techniques, we can controllably we can create the crop structures. So as we create these nanostructures, what we can do is we can enhance the material's property. For example, the graphene, it's because it's very thin and it won't observe only a small amount of light, for example, because it looks like very transparent, like you see here. But as we create these nanostructures, this material can observe more than, well, order of magnitude of light. And we use these nanomaterials, structured nanomaterial graphene for photosensors. So these graphene materials can sense and detect the light based on the mechanisms, or when the lights are emitted on these materials, the electrons generated and the flow. And by detecting the number of electrons generated by these lights, and we can detect the light intensity. So how could we use these, well, structure and the materials? The mechanism is that we can, well, increase the areal density. So compared to the flat, well, graphene, as we create these wrinkles, then you can imagine as we observe the light on the structured graphene, we increase the area of observing the light. So that way we can increase the absorptions of the light, so that we can increase the sensitivity of light. Even we can synthesize the hybrid materials by combining these gold nanoparticles with the nanomanufacturing techniques. We can even, well, enhance the sensitivity of the photosensors. So that's what we do to modulate the materials property. And using these nanomaterials and the devices, what we can do, we can make the sensors that could be applicable for bio applications, for example. For example, we make these stretchable sensors. So these sensors could be wearable technique, well, platform to use for photosensors in a bioimplantable or wearable sensors. So these stretchable sensor, for example, I mean, wearable techniques uses the stretchability, for example. Usually the, well, conventional material like a silicon, and it's not, well, stretchable. So like I mentioned, that the traditional materials only stretchable within less than 1%. But, well, I mean, I suppose that when we have the device on our skin, the skin is stretchable to 15%. Like, so as we have the device, traditional device, the device and materials will fractures, but with this flexible device and based on these, well, highly mechanically strong devices, we can make the device stretchable and more conformal and highly sensitive, even when our body is, well, moving. So that's what we do. And even we can embed this in a contact lens, so that we can use these sensors as a platform for the, well, with the integrated systems with the smart lens system, for example. So also in our lab, we synthesize those materials using different techniques. So one of the, well, I mean, techniques we can synthesize the graphene material is the laser. So I mentioned that the CVD technique. So with the high temperature thousands, we can create the hexagonal bonded carbon atoms. But, well, the CVD techniques takes a long time to synthesize materials, up to 4 hours, and then but we can make, like, well, large scale. But using these other techniques with the laser, we can synthesize the graphene materials in a very short time scale, like less than a minute. And we can also fabricate the device. Usually the graphene requires us to transfer to make the devices on a different substrate. But using these laser techniques, we can synthesize materials in a very fast time. And at the same time, we can fabricate the device using these printing methods. So this laser allows us to synthesize the material simultaneously. We can fabricate the device. So this is how it looks like. So we use the polymer to synthesize materials as opposed to metal catalytic films that we use to synthesize the graphene using the CVD techniques. We use the polymer materials as a precursor. Well, the polymer that contains the carbon atoms, and we can use that polymer as a precursor to synthesize the graphene. So what happens when we radiate these lasers, and then those molecules and then elements like atoms, other elements other than the carbon evaporate, but the carbon atoms forms the hexagonally bonded atoms with the pore structures. So this is how it looks like. What happens when we shine the light on the polymer materials. So these, in a very short time scale, the carbon atoms form the hexagonally bonded atoms with the pore structures, and these shows the graphene-like structures. So even we can synthesize the hybrid materials of the nano particles using these techniques. What happens is when we shine the light on this polymer that contains the precursor of the carbons, at the same time these nanoparticle precursors, and we can synthesize these nanoparticles embedded in the porous graphene structures. Using these hybrid materials, what we do, this can be used, hybrid material can be used for broad applications. You can think of the energy harvesting or solar cells, or even catalysts to use for the hydrogen detections, and there's bioimaging and sensing. So here what we do, we will make the hydrogen sensor. So you can see these sensors is highly flexible, although this has hybrid structures of the nanoparticles and the porous graphene. This material is highly mechanically reliable to these bendings and the twisting. The hydrogen sensing is reliable on the bendings and the twisting, and then it shows a reliable and consistent sensing performance. It's because of the material's reliability. So the fabrication techniques we use, using the laser manufacturing, so we can fabricate the hybrid materials with the high mechanical flexibility and reliability, because those materials shows the coherent bonding between those carbonates and the nanoparticles. And this is how we can use those hydrogen sensors, because this hybrid material shows a high mechanical flexibility and robustness. We can use these materials for wireless sensing platform. So here we integrate these sensors with the wireless and then signal processing models, so that we can monitor any dancing leakage, especially the hydrogen leakage, for useful applications of the hydrogen vehicles and hydrogen storage systems. So we are monitoring the leakage of the hydrogen using those mobile phones, and we can monitor the concentration of the hydrogen, which is these our hydrogen hybrids based sensing systems. And just to give you a sense of what we do, even with the atomic lithium materials, we can modulate the fracture modes. And here we, with the collaboration with the other groups in University of Illinois, we explored that with using these atomic lithium materials, even we can modulate the fracture mode of the metal films. These metal film is widely used for biomedical applications, for example, like multi-electrodes. So it can be used for implantable the electrodes. But here, using atomic lithium materials, we can modulate the fracture mode. You can see as we add the graphene layer on the bottom of the metallic film on the flexible substrate, we can modulate the fracture modes. And then as we add this graphene, turns out that we can modulate the fracture mode, and we can extend and enhance the conductivity of the material. So as opposed to the bare metals, and as we add this, just one thin sheet of the atomic lithium materials of the graphene under the bottom of these metallic films, we can enhance the conductivity and the mechanical robustness of the material. So this shows that how we can modulate the fracture mode, and using these the physical mechanisms of modulating the fracture mode. We use this as a universal tools to enhance the electrical properties and then mechanical roughnesses of the film. So we integrated our approach with these flexible displays so that we can enhance the mechanical roughness of these flexible display. So with that, I'd like to just stop here. So I hope that I just gave you the sense of how we can use atomic lithium materials for quantum materials or the quantum sensing. So with that, I'd like to stop here. So if you have any questions, I'll be happy to answer.
Thank you, Pilgyu and Rob. Great talk and like the sense that you gave us for the what you guys do in the lab. We're a little bit behind of time, so maybe we can take 2 questions and then we can move forward. Any questions, please unmute yourself and ask. Thank you.
Hi, Dr. Kang. This is Moti speaking. Great talk, by the way. Hi. And it's really interesting, the part you were talking about, the flexible. Hopefully we can do the flexible electronics sometime after.
Sounds exciting.
And I just had a quick question about the part you were talking about, crumpled graphene sheets and how they can increase the area when you shine the light. I was just wondering, did you start with cylindrical graphene sheets too? Is it possible, or I was just thinking maybe that would be easier in terms of synthesis rather than crumpled nanostructure. I mean, I just wanted to know what would be the difference between the cylindrical or crumpled structure.
So the cylindrical, so here the structure is 2-dimensional, but as we created the crumple structures, I'm not sure what you mean by the cylindrical structures, but
I mean, when they have like those single wall, for example.
Oh, carbon nanotubes?
Nanotubes?
Oh, I see.
Yes, something like nanotubes, yes.
Oh, I see. So here, the carbon nanotubes is one of the, the configuration of the carbon crystal structures. But here we use the 2-dimensional graphene. So instead of the 1-dimensional graphene, the people consider the carbon nanotube is 1-dimensional because it's long. But here we use the 2-dimensional materials. But as we create these 2D structures, 3D-dimensional structures of the wrinkles, I would say the crumples, we can create, increase the area. For example, here, this image shows that, it gives you a sense of how we can increase the area here. So as we create the wrinkles, you can imagine compared to the flat graphene, these has a higher, absorption area. So as we have more number of crumples here, as we create these crumples, and we may have higher areal density. So that's how we can use actually increase the area using the pore structures. So as we do the collaboration projects of using the pore graphene, even we create even way higher area. So pore structures allows to have a higher surface area of the graphene, but here we use the wrinkle structures.
I see. Okay, great. Thank you, Dr. Kang.